The title of our webinar is Faster Supply of Tailor-Made Biocatalysts Using Automation Technologies. So tailor-made biocatalysts are essential for bioeconomic strategies worldwide. They offer enormous flexibility and can be adapted to many different applications. However, developing them often requires substantial time and manpower. To address this challenge, the AutoBioTech, this is the automated platform which has been established at Forschungszentrum Jülich, Germany, as part of the Jülich Biofoundry. So I'd like to introduce our guest speaker, Tobias Michael Roche. Tobias Michael Roche is a PhD student at Forschungszentrum Jülich in Germany with a background in molecular biology, in molecular and applied biotechnology from the RWTH Aachen University, specializing in bioprocess engineering. In his PhD research, he applied his experience in process automation and molecular biology to push the limits of high throughput biocatalyst production. As he is a lead responsible for the AutoBiotech platform within the Jülich Biofoundry, he is helping shape the future of automated biotechnology. In this presentation, in this webinar today, Tobias will showcase how AutoBiotech is transforming the biocatalyst development by harnessing the speed, precision, and reproducibility of automated technologies. From single enzymes to fully engineered bacterial strains, the team are enabling high throughput construction at an unprecedented scale. You'll see how essential devices, such as our Certus Flex dispenser, work together, bringing their unique capabilities into focus. Tobias will also give us examples and highlight the power and promise of automation in modern biotechnology. So over to you, Tobias.
Thank you very much, Sarah, for the kind introduction. And exactly with that, also a very warm welcome to me, to everyone in the audience. And I'd like to straight jump into our talk or webinar today. Now, first of all, although Sarah already mentioned it, my name is Tobias Roche. I am a PhD student at Jülich Biofoundry with a biotechnology background. And just to tell you where I'm actually holding this webinar from, we are located close to Cologne, Aachen, or Düsseldorf, so somewhere around here on the map, very close to the Netherlands, Belgium, Luxembourg, and even France, so quite a bit of countries available around. More exactly, we're part of Forschungszentrum Jülich. I really apologize for that horrible name for English speakers, which is a very large research complex with a few thousand employees. And more specifically, Jülich Biofoundry is part of the IBG1, or Institute for Bio and Geosciences of the Forschungszentrum Jülich. As we heard already, Jülich Biofoundry is all about biocatalysts. Now, biocatalysts can be a few different things, one being whole cells, where we use them for growth-coupled production processes, meaning that we use the cell and its whole metabolism as a catalyst. The second big option is single or multiple few enzymes. These can be used for biotransformations from one specific substrate, which is not necessarily a carbon source on which an organism can grow, into a new and useful subproduct. In between, there's something called whole cell biotransformation, where we create cells that harbor a lot of a single or few enzymes, and that way also allow a biotransformation without a lot of purification. One could say that biocatalysts are basically a chemical toolbox for industrial biotechnology. And this is exactly why at Jülich Biofoundry we want to tailor these in order to achieve new things. We do this, for example, by enzyme engineering, optimizing protein expression, metabolically engineering host organisms, or even performing adaptive laboratory of evolution on the host. In order to do this, we have quite a few different technologies at Jülich Biofoundry, and I'd like to present our three pillars or key technologies. First of all, we have to engineer our biocatalyst in the very, very small scale. And for this, we have the already mentioned AutoBioTech platform, which is an integrated work cell for all steps necessary. Strains which either are or produce biocatalysts then go into a screening process. And this is done in our mini pilot plants, which are scalable cultivation and liquid handling platforms shown in this picture here, which allow us to screen dozens of strains at once. Finally, strains which show promising results in this stage then go into a scale-up, since this is really important for industrial biotechnology, as usually things are produced in the cubic meter scale instead of the milliliter or liter scale. And as a first step towards this point, we use our auto STR system, which consists of stirred tank reactors in the 1 liter format that connect to automated sampling into our liquid handling platforms. The workflow at Jülich Biofoundry begins in silico, meaning at a computer where we perform things such as metabolic modeling or key performance indicator prediction, and this allows us to create a kind of engineering blueprint for our biocatalyst. This blueprint can then be translated into pipetting instructions, which first of all go into the AutoBioTech platform to conduct steps such as DNA assembly, transformations, plasmid purifications, colony plating, picking, and so on and so on. Basically anything molecular biologists can imagine. After that, the strains which come as cryoconserved cultures go into a screening process in our mini pilot plants, and we quantify things such as final cell dry weight and product titers. The best strains, like I said, from here go into the auto STR system for further in-depth analytics, and this gives us a lot of information on the final bio process coming from these top performers. Of course, this whole workflow is a bit too large to cover in a single webinar, so therefore today we're focusing on the AutoBioTech platform, and we're going to look at how devices work in there. Like I said already, the AutoBioTech platform is an integrated work cell, meaning that it connects a lot of different devices with a single gripper arm, and it has defined boundaries shown in this diagram here. The boundaries mean that it's actually encased with a set of doors which protect humans from any malfunctions, which of course don't happen, but we do need them for regulatory reasons. And nevertheless, this whole setup allows the system to work peacefully and calmly without any human intervention. The AutoBioTech platform specifically consists of quite a few different devices, some of them connected directly to the spinnaker mover, which are a plate photometer, a thermal cycler, a liquid dispenser, foil sealers and peelers, a flow cytometer, as well as some incubators. The central mover also connects to a sophisticated liquid handler, our Tecan Fluent, which itself has 3 different arms for gripping and pipetting, as well as can access a few different dedicated devices, which are a centrifuge, an electroporator, a shaker heater module, a colony picker, and a pressure module. As you may notice, this is a quite complex system, but automated devices by themselves really aren't that intelligent. So in order to actually harness the potential of these devices, there is a lot of engineering that we've done in the last 5 years. And today I want to give a few examples of which processes now run on this work cell, how the workflows actually look, so which device performs which task in a process. And I also want to go into a few highlights where you will see what challenges may arise during automated workflows and how we actually tackled them with our automated devices. The 3 case studies I'd like to go into today are, first of all, the transformation of Corynebacterium glutamicum, where we look at reproducibilities. Secondly, a strain library screening process, where we will talk about scientist time usage. And finally, a high-throughput screening campaign, where it's all about parallelization. Jumping into the first case study, I'd like to highlight that Corynebacterium glutamicum is our pet organism. We use it a lot at our institute, and it's an industrial platform organism that can be used for a variety of production processes. We have to transform this organism in order to teach it new biocatalyst properties, and this means that we introduce heterologous DNA via electroporation. This is a physical transformation method which is not really that easy to automate, And this is also why we are today talking about a throughput of around about hundreds of strains per week. The workflow for transformations of Corynebacterium glutamicum is shown on this slide. The workflow is rather simplified as it is very lengthy, and if I added all liquid handling operations in between, this would not actually fit on the slide. So please just imagine a liquid handler or dispenser at every arrow shown. The workflow begins with a set of two pre-cultures of our organism, a main culture in special medium and resuspension in a competency-inducing medium to create competent cells which are ready to take up DNA. After that, the cells can be combined with DNA and actually electroporated to introduce this new DNA. They are heat shocked in a thermal cycler, a recovery takes place in an incubator, and finally, the liquid handler performs the plating steps in order to turn the freshly transformed cultures into single agar colonies. Again, this workflow is rather detailed, and therefore I will today only go into two examples or challenges, and those arise at the cultivation stage and at the electroporation stage. First of all, regarding the cultivation stage, a challenge can be supplying large liquid amounts to a liquid handler or generally an automation system. Normally, we would do this directly in our liquid handlers with 100 milliliter reservoirs from where you can easily pipette any aqueous solution as you can see here. However, problems with liquid level detection and manual filling requirements occur when we try to conserve our liquids. Because when we pipette liquids back into the reservoirs, as pipette tips often have excess liquid, this causes foaming and may lead to aspiration of air instead of liquid. The result is that no culture medium or recovery medium may enter a certain well, leading to direct failure of a replicate without any real reason for it. We recently were able to actually tackle this problem with the Certus Flex liquid dispenser, which can very precisely and automatically dispense liquids, meaning that we know exactly how much volume enters each well, and therefore all following processes work perfectly. This does not only give us consistent volumes, but also more autonomy, since the reservoirs shown in this video at the top have to be filled manually before every operation, but Certus Flex can actually take bottles, which are connected to the device once a day, a week, depending on the liquid usage. The second challenge that I would like to show for the transformation of C. glutamicum is fine-tuning pipetting for electroporations. With standard liquid handling, both for humans and robots, it occurs that we create bubbles, as we saw on the last slide. These bubbles actually are also a problem for electroporation, since they can lead to an uncontrolled electric discharge called arcing. We found that with the standard liquid handling protocols, arcing occurred in 65% of our wells, and this was a big problem because it meant that reproducibility pretty much was down to only 33% across a plate, which is not acceptable. However, by completely eradicating leading air gaps in our pipetting scripts, we were able to completely prevent bubble formation and therefore also reduce the arcing rates down to zero, which really increased our reproducibility on a technical side as well. Therefore, we saw that automated devices can really be tuned, or you can optimize the ones you use for operations in order to get the exact reproducibility that you need. Next, I would like to go into an example on strain library screening. In this example, library means a broad set of strain variants, and these strain variants are different microbes which have mutations either in their genome or on a plasmid, and these mutations will lead to different biocatalyst properties. In order to find the best variants in such a screening process, we measure key performance indicators such as enzymatic activity, or even the growth of a certain strain, as of course, we can only produce a biocatalyst if it's also self-replicating. We quantify these key performance indicators by photometry, since this gives really fast results and is also directly available in the AutoBioTech platform. And we're again talking about hundreds of clones per week although this may also actually reach the thousands, depending on the exact campaign that is being done. The workflow for strain library screening looks a lot more simple than for the transformation. This is because the molecular biology in this case may be done manually, and the library can be created in a rather random one-pot reaction, and the colonies are all plated at once on one or multiple agar plates. These agar plates enter a liquid handler together with a plate filled with our dispenser, and the colonies are picked into the liquid medium. These liquid media cultures are then cultivated, and this is where a very variable part of the workflow begins, as either we will only cultivate the cultures, or we may also induce them in between, add an assay reagent, add new medium, or feed a certain metabolite. In the end, there will always be the readout, and the two challenges which I would now like to look at with you are the picking and the reagent addition. Picking colonies is a very tedious and sometimes also error-prone task when you want to make sure to not pick replicates and also to always pick only one colony. However, in order to take some strain off the scientists, we at Jülich Biofoundry have a Piccolo module for our fluent liquid handler, which can actually do this automatically as shown in the video. Specifically, the device uses a light table shown here in conjunction with a camera mounted at the top in order to detect colonies and that way receive coordinates where they can be picked. This works at a rate of round about 800 colonies per hour, which in fact, at least in our experience, is similar to the speed of a human scientist. And therefore, one could ask the question what the benefit is. Well, it's exactly the walkaway time. The walkaway time already exists with round agar plates, as the picking of a lot of colonies from one plate takes some time, but this really reaches a new level when we use SBS formatted trays, meaning rectangular trays in the same form as a microtiter plate, which can be gripped by every part of the AutoBioTech platform, and therefore the whole process can work by itself. Finally, well-to-well picking is also something that is really enabled with the system, as we can make sure that when we spot our colonies, instead of plating them on a whole plate, we will have spots ranging from A1 to H12, just as on a microtiter plate, and the robot can ensure that a colony from one well always lands in the same well in the medium plate, and therefore we get intrinsic colony lineage with less amounts of sample tracking being required. The second challenge that I wanted to highlight is the addition of media, or generally how we can save more time in this variable part of the workflow. The first device I'd like to highlight here is our plate reader, the Synergy H1, which has a lot of data export options to create the data in a CSV file. And this CSV file can actually be read on the fly during a workflow, meaning that we can evaluate where our experiments are at during the run of the experiment. In conjunction with the Certus Flex, which allows us to generate dispensing instructions as an XML file, which is both human and computer readable, we can actually make sure that decisions that may arise from data here gets into the dispensing instructions for the Certus Flex, and therefore makes it possible to give every well the exact amounts or types of liquids that they need at a certain time point. One could even do something as complex as drawing our initials here, but that really isn't necessary that often. Overall, these two devices together allow automated decision-making on a single well level, and therefore take a lot of work away from the scientists so that they don't have to wait for data and manually make their decisions, but instead the system does this by itself. The last example I will be looking into today with you is high-throughput screening. In this context, high-throughput screening again means a measuring of key performance indicators. However, instead of microbial strains, I will now be talking about single enzymes and therefore specifically new catalyst-substrate combinations. The real challenge in such a workflow is exact timings, as the enzymatic assays that we're using here are very precise and therefore may change a lot depending on the exact incubation times. The throughput in this case definitely is in the thousands of reactions per week and basically is limited by the amount of samples that we actually want to measure. The workflow for this type of experiment looks again rather simple, with an empty plate being filled with buffer by the Certus Flex. The fluent then adds substrate to this buffer. The Certus Flex again adds enzyme, and after a sealing of the plate with a plastic foil, it's incubated for a set amount of time. And after that, the assay reagent is added, incubated again, and the final readout occurs. While this seems very simple, the data from this is actually quite complex. An example is shown here where a total of 95 substrates were tested, again 37 enzymes, and you can see from the red boxes in the heat map that quite a few hits were found and a lot of these actually weren't known before. The heat map here totals to 10,500 reactions, which were conducted in 2 weeks, and this really showcases how well the AutoBioTech performs in such an experiment. Now let's look at the exact timings. When a human scientist performs this workflow, we start with a 2-hour preparatory step, which cannot actually be automated, but all following steps take round about 2.5 hours to complete, with the dark gray boxes in this diagram being the steps conducted by a human, and only the light gray boxes being ones that are actually automated. However, like I showed already, our automated devices can help to really perform the same things and also accelerate this workflow, resulting in this new Gantt chart shown here. As I said, the preparatory step stays, so these 2 hours cannot be omitted in this case, but the other operations, which before stretched all the way to this red box here with the manual end, now are condensed down from 2.5 hours to just over half an hour. This doesn't only make the assay a lot faster, but it also allows us to do something called parallelization, where once one of the plates is incubating, we will start processing a new plate. So not only does this whole setup give us more reproducibility because there's little human error involved, we also get scalability because we can really parallelize the processes for different plates and actually stack this more than 2 times shown here, but instead up to round about 12 times. Also, there's a lot more efficient time usage here for scientists, since, as you can see from the balance of dark and light gray bars, there is nearly no operation which the scientist has to perform, but the preparatory step and the swapping of enzymes in this case. The last point I would like to talk about today is the handling of non-aqueous liquids, as this is actually also really important for enzymatic assays sometimes. The first challenge with non-aqueous liquids can be volatile solvents, as these like to evaporate. Especially in reservoirs at the ones shown at the beginning of the webinar, you will get a lot of evaporation, and this is where a bottle supply, as from the Certus Flex, really comes into play, as it keeps volumes and concentrations constant. Secondly, pipetting accuracy can really be different depending on the surface tension and the viscosity of a liquid. Just like human, a robot has to learn for every single liquid how it actually pipettes that, meaning that for every liquid, we have to define a liquid class which has a lot of different parameters. However, when we have the option to do a gravimetric calibration, as with the Certus Flex, we get inherent accuracy for every liquid from a single standardized calibration procedure, which really is a lot easier than making liquid classes and therefore takes some work away from the automation engineers. Lastly, some liquids, also aqueous ones, will perform material degradation depending on what materials you're using. Solvents, acids, and bases can sometimes deteriorate plastic tips, which are usually used in liquid handlers or in manual pipetting, which can lead to contamination of samples. This is again where things like steel valves, as in the Certus Flex, really give elevated robustness and prevent our experiments from being contaminated with compounds that we do not want in there. Therefore, for handling non-aqueous or for very differently behaving liquids, we really prefer a dispenser. And if you can choose to use a dispenser, I would advise you to do so. But of course, we also know that there are cases where the pipetting is too complex to do that, and then we have to find other ways. With this, I would also already like to come to the end of the webinar and finish with 3 take-home messages. The first take-home message is that specialized devices are really something to look out for. Executing one task with one device perfectly is by far better than having a device which performs a lot of tasks mediocrely, because this one task that's perfect will definitely not be a problem in your workflow and just increases reproducibility by a lot. Secondly, integrated workflows are a game changer when it comes to walkaway times for scientists. An integrated platform will allow the scientist to start an experiment and come back for the end, giving more time to let automation work for you instead of working for automation where people will sometimes be standing next to a liquid handler all day. Finally, parallelization is something one should really consider when it's possible with a workflow. Parallelization allows to scale workflows and therefore make the most of a single automation system without having to perform any further investments at the same time. And with this, I would like to already end the webinar and thank you very much for your attention. Do feel free to ask any questions after this, or also email me, write me at LinkedIn. And you may also want to check out some of the publications from the AutoBioTech platform, such as the first one shown here, which actually shows all technical and some workflow details, as well as some of the off-spinning publications, which show some real-world research examples. Finally, I would like to thank Nano very much for hosting today's webinar, and I am now open for any questions.
Thank you so much, Tobias. That was a very informative and engaging presentation, very clear. If anyone has any questions, please use the Q&A box down below and you can ask questions. We do have a few questions that have come up. So let's have a look. And as Tobias mentioned, you can always send in the questions later. So the first question. How did the use of the Certus Flex influence the speed of your experiments in comparison to other liquid handlers?
So that really depends on the operation that you're performing. Generally, I feel like the Certus Flex really benefits from either very complex but few liquid containing steps, or from very simple steps. So for example, the Certus Flex is a lot faster at distributing a single liquid across a plate than our liquid handler, simply because it doesn't have to do any aspiration and dispensing operations, but instead just processes one well after the other. The same goes for mixing up to 8 different liquids at the same time, where it's really a lot faster because it just has all these channels available to use at once to dispense liquids, while a liquid handler usually only has up to 8 channels and has to use them one by one to dispense. However, of course, if you want to do something such as moving liquids from A to B, this is something where you will need a liquid handler. So this really depends on the type of operation that you're doing.
Yeah. Okay. What about sterility? So we've got a question about sterility with media connected to the device. Any concerns there?
Actually, no. So the very nice thing in the AutoBioTech platform is that we only work with bacteria, meaning we have no eukaryotic cell culture and also rather little other eukaryotes. And therefore, yeah, we use a lot of antibiotics and don't have any problems with sterility. I myself did a test on sterility for the Certus for our platform and found that you can easily leave a bottle of medium connected for up to 14 days, depending on the medium itself. and you will not get any contamination in the medium. And if you spot the medium onto an agar plate, you will also not see any colonies or yeah, fungi growing.
Mm.
So that's really an important point for us. So we don't have to fill the media every day, but instead we connect them once, and then they're fine for all workflows coming down the line.
Okay. Um, how do you clean your valves? What's your cleaning procedure?
Well, that depends a lot on which liquids are used before.
Yes.
If it's actually just clear water or buffers that may have even been filtered sterile for some reason, there's
Mm.
not really any cleaning I have to do except flushing the valves with water, maybe 70% ethanol or something like that.
Yeah.
When we use something like complex media, which will often come from, for example, yeast extracts or something,
Mhm.
these may sometimes contain larger aggregates of some kind and may sometimes clog the valves. In that case, we use the cleaning kit that's available to flush the valves a bit more thoroughly, dries them, and then they're ready for the next go again. But that's really an iterative cycle to find out how much cleaning which
Yeah.
liquid requires.
Okay, good. Let's see. Let's see. Okay, I think oh, hold on. Uh, what was the main reason why you swapped to the Certus Flex for your application?
Well, the main reason was that first of all, we wanted the elevated autonomy. So with the liquid handler and the reservoirs, there are many different workarounds to store liquids in a liquid handler, such as having a cooling carrier where the liquid is cooled. You can try to seal them with foils, or you may supply them in such large amounts that they're always present. There's also things like pumps, but all these options either weren't applicable for our liquid handler,
Mm,
or we didn't feel like they were worth the amount of work you have to put in to make them work. And therefore, the autonomy that the Certus Flex gives us, where you just connect a bottle and the system can really simply tell the software, I want liquid A now. That's just a big game changer for all procedures, such as the competence cells, where we now don't have to fill plates manually beforehand, but instead the plates are filled really on the go. And therefore, we also waste less material when we're actually testing
good.
or when workflows crash, because
Yeah.
instead of having everything prepared every time, we can instead let the system prepare everything as it goes on. And
Mm,
therefore, if we don't reach a certain point, it's also not going to have prepared that. The liquid is not going to have been dispensed, so it's not wasted. And also, the plate that is going to be touched will not have been contaminated with anything.
okay. We've got a question. Are you familiar with the mantis, Tobias?
Yes, I am familiar with the Mantis. That was definitely also in the selection group when we looked into dispensers. I mean, there is quite a few on the market. But the thing which was really important for us as a publicly funded institute is that we can't have too many running costs.
Yeah.
So with devices like the Mantis, or there's also other manufacturers around. Many of them have a lot of parts which have to be replaced every week or every liquid. And this creates rather high running costs, which are really hard for us to tackle. This may, of course, be different for other customers, such as ones from industry, where this might not be a problem. But again, the fact that we don't really have to do any maintenance on the device and the valves just work all the time makes it a lot easier to set up the device or keep it set up instead of having to change a cassette every day or just having the whole plastic waste that also
Yeah.
comes with these other devices.
The question was, would a mantis be capable as the Certus? So, yeah, it would be capable, but there's lots of added.
Also, I think the Mantis, when I remember correctly, has a quite different principle. The volume ranges that you can supply are fairly different. That's actually also something that was important to us, that we wanted a device where you could connect at least 250 milliliters, which is possible with the first Certus.
Mhm.
But most other devices on the market, such as the Mantis, don't actually allow that.
Yeah.
And that's why we chose the Certus.
Okay. Good. And you heard that from him, not from us.
Yeah, that's true.
Okay. And with that, if there are no further questions, I would just like to again, once again, thank Tobias for an excellent presentation and for the webinar. N-Nano and Future Drop Fritz Gigerage, we will be at SLAS in Boston next month. Tobias, are you attending any shows or conferences? Are you doing any talks any time this year?
Sadly, not SLIS. I will be at a few German ones,
Okay.
such as the Himmelfahrtstagung, in case anyone knows that. I will try to make it to SLAS Europe in Vienna, but that depends a lot on money situations. And I may be also at some biotech summits down the year, but that's to come. If anyone wants to meet me, feel free to follow me on LinkedIn because I'll definitely be posting where I'm going there.
Perfect, perfect. And again, once again, reach out to us if you would like any further information from Tobias or ourselves. And thank you once again for joining us at this webinar. Have a great evening. Thank you. Bye bye.
Bye.